Hello, for today we'll be talking about gene regulation in prokaryotes. Our stomach cells produce hydrochloric acid to break down food, but this does not happen in ice. Despite this, both our eye and stomach cells possess the same DNA, which is present in all cells throughout the body. However, certain portions of this DNA need to be regulated in order to prevent the production of hydrochloric acid in eye cells. In this case, genes should be regulated and controlled. Now let's proceed with the characteristics of prokaryotic DNA. Prokaryotes possess circular double-stranded DNA, lacking histones and introns, and are distinguished by the absence of a nuclear membrane. Consequently, transcription and translation can occur simultaneously, leading to a more direct and tightly controlled gene expression in prokaryotes. This is due to DNA not being separated from the translational machinery in the cytoplasm. Additionally, DNA in prokaryotes is not enclosed in a distinct compartment but is freely accessible to the cytoplasm. Prokaryotes also exhibit polycistronic mRNA, where a single mRNA encodes multiple separately translated proteins. Gene regulation is the control of protein generation or RNA product from a particular gene. It has two types. First, the repressed gene regulation, which decreases gene products, and second, induced gene regulation, which increases gene products. So the main goal of this process is to allow cells to save energy and nutrients, since product is only produced when needed. We have a feature that allows protein synthesis to be controlled coordinately in response to the needs of the cell, and this is what we call the operon. So an operon is a cluster of genes that are transcribed together to give a single messenger RNA molecule, which therefore encodes multiple proteins. So an operon is made up of the promoter, additional sequences that control the transcription or the operator, and the structural genes. We have two types of operons. Inducible operons are those whose transcription is usually off and needs to be turned on. On the other hand, repression operons are those whose transcription is normally on and needs to be turned off. Now let's discuss negative inducible operons. In these operons, the operator site exerts negative control. In this case, transcription is inhibited by the molecule that binds to the operator. These operons are typically inactive and require activation to initiate transcription. Additionally, inducers, which are small molecules, play a crucial role in activating transcription. When inducers are absent, active regulation protein binds to the operator. This inhibits the binding of the RNA polymerase to the promoter, so no mRNA products are formed. In this case, the binding of the active regulation protein to the operator prevents transcription of the structural genes. On the other hand, when the inducer is present, it binds to the regulator, thereby making the regulator unable to bind to the operator. In this case, the transcription takes place. Now let's proceed with the negative repressible operons. So these operons, their control at the operator site is negative. However, despite the usual state of transcription being on, it needs to be switched off, making it repressible. So we have co-repressors, which are small molecules playing a crucial role in this process by binding to the repressor and enabling it to bind to the operator, thereby halting the transcription. Now let's discuss what happens when no product U is present. Number 1, regulator protein is an inactive repressor unable to bind to the operator. So this enables the RNA polymerase to bind to the promoter and initiate transcription. Transcription of structural genes takes place then. The mRNA transcript is translated into the precursor T protein, and this protein acts as a coenzyme in the biochemical pathway, converting precursor T products into intermediate products. So these intermediate products are converted then into final products. Following this, the levels of product U or the co-repressor builds up. Because of this, product U binds to the inactive regulator protein, activating it. This halts the transcription. Now let's proceed with the positive inducible operons. So in these operons, transcription is off. A substrate used by the enzyme to produce a product in the metabolic pathway binds to the inactive activator and activates it. The activated activator binds to the operator, which enables the binding of the RNA polymerase to the promoter to initiate transcription. Transcription will then be on by this time. On the opposite, positive repressible operons have transcription turned on. To turn this off, products bind to the active activator, inactivating it. The inactive activator cannot bind to the operator region. As a result, the RNA polymerase cannot bind to the promoter region and no transcription will take place. Now let's discuss some examples of these operons. The lac operon of E. coli is an example of a negative inducible operon. The repression of the lac operon never completely shuts down transcription, but it only slows it down. The following structures make up the lac operon. First, the inducer or the allolactose, second, the repressor encoding gene, third, the promoter, and fourth, the operator. This operon also encodes for the following structural genes. For lac-Z, it encodes for beta-galactosidases, lac-Y encoding permeases, and lac-A encoding for transacetylases. In the absence of lactose, the active regulator protein binds to the operator, so RNA polymerase then cannot bind to the promoter. This inhibits transcription and no transcription will take place. When lactose is present, some of it is converted into allolactose, which is an inducer. This binds to the active regulator protein, making it inactive. This inactive lac repressor cannot then bind to the operator region, so RNA polymerase then can bind to the promoter and initiate transcription. Therefore, the structural genes are transcribed and translated. Let's proceed with catabolite repression. So this employs positive control mechanism, which utilizes glucose in the presence of other sugars, effectively repressing their metabolites. The activation of this positive effect is facilitated by the catabolite activator protein, which binds to CAMP, forming a complex that then binds to the site located just upstream from the lac gene promoter. When glucose is low, the levels of CAMP are high. CAMP binds to Cap and this complex binds to the DNA. This enables the RNA polymerase to bind to the promoter and initiate transcription. The results are high rates of transcription and translation of the structural genes. This produces glucose from lactose. On the other hand, when glucose is high, the levels of CAMP are low. As a result, CAMP is less likely to bind to Cap. RNA polymerase then cannot bind to the promoter region of the DNA as efficiently. In this case, transcription rate is low. Let's now proceed with the last operon. TRP operon is an example of a negative repressible operon. In this operon, we have 5 structural genes, TRPE, D, C, E, and A. These 5 enzymes together convert chorismine to tryptophan. When tryptophan is low, the TRP repressor is inactive and therefore cannot bind to the operator region. RNA polymerase then can bind to the promoter region and initiate transcription. Therefore, transcription takes place. When tryptophan is high, tryptophan binds to the inactive regulator protein and activates it. This enables the binding of the repressor to the operator region. Thus, transcription is then shut off. This ends our lesson. I hope you have learned a lot from my lecture. Thank you.